So we're going to sort of finish up movement today, talking about the cerebellum. And if you remember, we were going to start with the cerebellum, and I'm really glad we waited till the end because I'm not sure you'll really be able to wrap your head around it all, because the cerebellum is going to tie a lot of things together. And so hopefully it'll work today that as we're talking about the cerebellum and its loops and its circuits, that we're going to be referring to a lot of the motor pathways that we did like 2 weeks ago, so that you're going to understand its influence on sort of this overall concept of movement that we've been talking about for the last couple weeks. And this is a quote out of, not your textbook, something else, but it says, "The most difficult problem of all is that the cerebellum is an essential node in numerous feedback pathways. Hence, studying the cerebellum in isolation from other components of these loops reveals little about its true function." So again, I think we have to think about the cerebellum in the context of what we've already learned about motor pathways and about movement execution. So today we'll start out with just talking about the general cerebellum functions. We'll do an overview of the anatomy, and then we'll do some circuits. We'll talk about input and output. The nice thing about the cerebellum is that a lot of the places that it gets input from, it gives feedback to, so we have this sort of circular pattern. Plus it gets input from a couple other places as well, so we'll talk about those. And then in the afternoon we'll talk about cerebellar dysfunctions, what that looks like, and we'll focus on a couple conditions where the cerebellum is affected. So when we talk about just general motor functions of the cerebellum, there are motor and non-motor functions. We're going to primarily be talking about motor today. And the cerebellum, it does not give us any direct projections to the spinal cord. So it's not executing direct movements like the motor cortex does. But what it does is it serves to help as sort of a feedback mechanism to the cortex to say, tell me what your plan is. I think maybe you need to tweak it a little bit. Now do it. And so, and it provides sort of a postural set or a background of information on which the motor cortex can make decisions. So it helps to, in the end, smooth out movements to coordinate multiple joint movements at the same time, so that movements aren't broken down into, well, first my shoulder moves, then my elbow moves, then my wrist moves. So it adds synergy to movement. You'll hear that term, particularly when we talk about this synergy in the afternoon. It allows for activation of antagonists and inhibition of sorry, did I say it backwards? Activation of agonists and inhibition of antagonists. We know that some of that happens at the spinal cord level, but it's getting input from the cerebellum that helps with that regulation. It's going to help to compare what we want to do with what actually happens. So for those of you that watched like the NCAA game, the championship the other night, you know that guy who botched the 2 free throws near the end of the game. Thank you, that made me lose my bracket. You know, clearly that was not what he intended to happen. He probably practiced that thousands of times. But again, it allows us to say, hey, that didn't quite work the way I wanted it to. Maybe I need to adjust and do it a little bit differently. And then it is going to provide input that's going to help to maintain our balance, our upright position against gravity, so our extension against gravity, and to maintain muscle tone and tension. So in just looking at what the cerebellum does, we know it doesn't directly connect to the lower motor neurons in the spinal cord, but where do you think it's going to connect to? If we're talking about balance and posture, what's it going to have connections with? Vestibular nuclei, the vestibular system. So we know we're going to see connections with the vestibular system. If we're talking about actual execution of movement, where is it going to have to connect to? What part of our brain executes movement? Motor cortex, and those in the premotor cortex areas, the SMA and the PMC. So we know we're going to, some of those connections we're going to see. We're also going to see some connections with other parts of the brain, but just to get you thinking about where the cerebellum is going to have an effect. The other thing that the cerebellum is helpful for, necessary for, is motor learning, and this is something that you have probably talked a little bit with Al about, either in the adult class or theory class. But when we start learning a new movement pattern, typically we have to break it up into component parts. And so, how many of you guys have learned a new skill, either like a sport or a musical instrument or something as an adult? Okay, so for those of you guys that have, think about for a second how you started, when you first started doing it. So I have two examples. I have tennis and guitar playing, are two things that I learned well after my brain had matured and all the synaptic connections were pretty much in place. And I can remember tennis that, like, I could only think about like one body part at a time. So, you know, if you're going to hit a ball, like, your feet have to be a certain way, and then your trunk has to rotate a certain way, and your hand has to be in a certain position on the racket, and then you're thinking about like your shoulder movement, your elbow movement, and your wrist movement. And I can remember when I first started trying to hit the ball back, that like the more I focused on, he'd say like, oh, your wrist is blah, 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 you know? He was foreign, I don't remember the accent. But you know, the more I focused on the wrist, the less, you know, then my feet would be out of position, or then I would be hitting too hard or whatever. And you really, in guitar playing, for those of you guys that have done musical instruments, like, you know, I'd have to focus so much on just going from like a C to a D chord, then I'd lose the strumming pattern with my other hand. It was like I could only do one thing or practice one piece at a time. So if you probably, those of you who have learned something as an adult can think about that learning process that happened, and most likely you had to do that chunking of the learning process until everything started to become more fluid. And it becomes more fluid with repetition of actions. The more you practice, the more you do it, the more automatic it becomes. It's because you're building those synaptic pathways, and a lot of that is happening at the level of the cerebellum. The cerebellum is going to start to connect those motor actions together so that you can have that synergy of movement, so that your shoulder, wrist, and elbow all start moving in the same way at the same time. The cerebellum will store that motor memory, and as you're doing it, as you're practicing, remember the cerebellum's constantly comparing, you know, how you thought that racket was going to swing, or how you thought that ball was going to go across the net, to what actually happened. So giving you feedback on, you know, that time you pronated too much and the ball went straight down, or you didn't pronate enough and the ball went way up in the air. And so you can adjust that movement the next time you do it. So again, the cerebellum is going to be key in learning new movement patterns and adding to the smoothness or the automaticity by which we can do them. So again, it's not telling us what to do, it's giving that feedback to the motor cortex and other areas of the brain to refine those motor patterns. Does that make sense? Okay. We're not going to talk about the non-motor functions of the cerebellum. It will come up when we talk about disorders, because when we have dysfunction of the cerebellum, we often see problems with things like memory, language, or visual processing, and we sometimes have issues with emotional regulation or behavioral regulation as well. The mechanisms, particularly the emotional regulation piece, are a little unclear, but the data shows that as the cerebellum, as we have decreased volume in the cerebellum or destruction of cerebellar fibers, we tend to have problems in these other areas. So be aware of that, but we're not going to be talking about the circuits for this or the mechanisms of this today. And we'll see as we start to do the circuitry that the way the cerebellum is able to accomplish, particularly the motor functions that I talked about, is that it's going to have reciprocal connections with the motor cortex. So the cortex is going to send the cerebellum information, the cerebellum is going to do that sort of compare, adapt, adjust, and send a new plan or a refined plan back to the cortex. So we have reciprocal innervations with the motor cortex. And the cerebellum is going to be highly an integrative structure. So it's also getting a lot of sensory information from the spinal cord, from nuclei in the brainstem, from areas of the cortex, and it's going to use that sensory information to do that motor refinement. So the cerebellum is going to integrate sensory and motor to help inform that motor plan. This is just a picture from your book. We're going to kind of get in more detail about this, so I'm not really concerned that you know this, but again, the cerebellum is not going to have output to the spinal cord. It will get input from the spinal cord, and it's going to have reciprocal connections with nuclei in the brainstem, and indirectly reciprocal connections with the cortex. So be on the lookout for that. We're going to talk about anatomy for just a little bit. This is a cartoon out of your book, and I don't like it for several reasons. I'll tell you why. But I put the picture in here anyway. First, just notice that when you look at this cerebellum, and you'll see the wet tissue and the models in lab today, that we have these folds. So the lines and the ridges, those are called folia, F-O-L-I-A. It's in your book. What this does, what these folds do, what did the folds do in the cerebral cortex? Increase surface area. So it's the same thing in the cerebellum, that the folia increase the surface area of the cerebellum, allow us to have more nuclei, more volume, more connections, and even though when you look at the cerebellum in relation to the cerebral cortex, the cerebellum looks much smaller, its surface area is about 75% of the cerebral cortex. So even though it looks about a third of the size or even a quarter of the size of the cortex, it has about 75% of the surface area. So it is a high volume, highly dense area, and cerebellum means little brain. So it's thought to be almost as important as the cortex, as the cerebral cortex itself. So when you look at the cerebellum, there are 3 lobes, and when we do the lab intro, I'll sort of orient you, but it's going to attach to the dorsal surface of the brainstem, so the colliculi usually sit just above it. And the reason I hate this picture is that it makes the anterior lobe look really big in relation to the posterior lobe. It's not. It's actually very small. So in this cartoon, the relationship, the proportion of the anterior lobe that you would actually see, so this would be a lateral view in this picture, the proportion of anterior lobe that you see is much larger than you'll see on any real tissue. So that's my main issue with it. So you have an anterior lobe, a posterior lobe, and then pressed against the brainstem, so this would be the anterior surface of the cerebellum, pressed against the dorsal surface of the brainstem, you have this lobe that stretches across the back, which is called the flocculonodular lobe. So it's shaped like this, but pressed against the back of the brainstem. The flocculus are the wings. The nodulus is in the center, and it's actually part of the center portion of the brainstem, which is called the vermis, and that's on the next slide. The flocculonodular lobe is the most primitive part of our cerebellum, and it primarily processes vestibular information, so it is sometimes called the vestibulocerebellum. It's going to be related to extension against gravity and eye movements, which makes sense with connections to the vestibular system. The anterior lobe is sometimes referred to as the spinocerebellum because it's going to be the primary receiver of spinocerebellar inputs, or inputs from the spinal cord. So remember, way back in week 3, we talked about the dorsal spinal cerebellar tract, cuneo-cerebellar tract, and the ventral spinocerebellar tract. Those are the tracts that went from the spinal cord to the cerebellum. They are going to project to the anterior, primarily to the anterior lobe. And then the posterior lobe, which again is in purple in this diagram, is the newest part of the cerebellum, so it's sometimes called the neocerebellum, and it's where most of our cortical inputs will be going. And so another term you might see for it is the cerebrocerebellum. So the part of the cerebellum that's receiving most of the input from the cerebral cortex. And I should say that this view is as if you were looking down on the brain, so it's a superior view. And again, I'll orient you when we do lab. This is another superior view. So this would be our anterior lobe, and then posterior. But this is another way to organize it. It's actually the more functionally sort of correct way to organize the cerebellum is into the central part called the vermis. And again, part of that vermis is the nodule, which is part of the flocculonodular lobe. On either side of the vermis is really easy to identify on the wet brains. If you take your index finger and put it on either side of that vermis, that's sometimes called the paravermal or the intermediate zone. So another term for paravermal is intermediate zone. So it's the area surrounding the vermis. And then everything else, both anterior and posterior lobes, that is on the other side of the perivermal region, are just called the lateral cerebellar hemispheres. So when we do, particularly the application lecture today, when we do circuits too, sometimes we'll talk about things projecting to the perivermal region, but we'll say of the anterior and posterior lobes. So that will be referring to this area in blue. We're talking about projections to the lateral hemispheres. We're talking about this area in green, and again, it would primarily be to both the anterior and the posterior lobes. Does that make sense? It's one of those things that, like, couldn't you pick one way to do it? No, they don't have one way to do it. You'll hear both lobes and sort of this organization used in terminology. Another part of anatomy that you need to be aware of are the deep cerebellar nuclei. There are 4 of them. They're grouped really into 3 different types. The most lateral and the most prominent is the dentate nucleus, which always to me looks kind of like what the inferior olivary nucleus looks like in the brainstem, kind of this tree-like structure. Bush. So that's here, there's the arrows. This whole thing is the dentate. Just medial to the dentate is the emboliform nucleus. So it's here and it's here. Again, you'll have some pictures in lab today too. Just medial to that are the globose nuclei, and they're usually a paired structure. Again, just medial to the emboliform nucleus. The emboliform and the globose nuclei together are called the interposed nuclei. And that's on your next slide, so you don't have to write that here. But just know that we typically don't talk about these as separate. We talk about them as the interposed nuclei, and we just use that term and go with it. And the most medial nuclei that are deep to the vermis are the fastigial nuclei. So there's one here and one here. The fastigial nuclei. So again, this cartoon out of your book shows you where they're located. The dentate is the most lateral, deep to the lateral hemispheres. The interposed nuclei, which are the globose and the emboliform, are deep primarily to the intermediate layer, the perivermal region, and the fastigial nucleus is right in the center, deep to the vermis. And you'll see that each of these have a function, separate functions, that their output is going to be to different regions of the brain. So they will be important when we do circuitry. I'm much less concerned with your understanding of the cellular aspects of the cerebellum. Your book spends a fair bit of time on this. The main thing that I want you to know is that there are 3 layers, and it's the Purkinje cells here, the big tree-like ones in red, that are the output cells of the cerebellum. All of the other fibers that you see here, the mossy fibers, the parallel fibers, they're integrative fibers. They work within the cerebellum to integrate sensory and motor information and to make those plans and do all of that stuff. But it's the Purkinje cells that seem to be most important for output. And in diseases like autism, the Purkinje cells seem to be the ones that are most affected. I think schizophrenia as well. So it seems to be these Purkinje cells that are most susceptible to damage. And there's no redundancy really, but they are the output cells of the cerebellum. If they get damaged, then the cerebellum can't do its job. And last anatomical tidbit of information, you guys have already seen cerebellar peduncles, probably more than you would like. You're used to this picture where we see the superior, the middle, and the inferior peduncles. What you've known up until this point is that they carry information to and from the cerebellum. Well, today we get a little bit more information about which ones carry what input. The good thing for the superior cerebellar peduncle is that it does not carry anything in, so you don't need to worry about what it's carrying in, at least not for this course. The middle cerebellar peduncle is going to be carrying the bulk of our information from the cerebral cortex. So information from the motor cortices, but also the sensory cortices as well. But again, from the cerebral cortex. It's going to do that through connections in the pons, and so that you sometimes hear the term cortical pontocerebellar tract, so it's going from the cortex to the pons to the cerebellum, but it's the pontine nuclei that project to the cerebellum and go through the middle cerebellar peduncle. So I'm going to do that 100 times today. You'll also get the tectum projecting through the MCP as well. The inferior cerebellar peduncle is going to be carrying information from the vestibular nuclei, also from the vestibular ganglion. What was the name of the vestibular ganglion? Scarpus, right? Spiral auditory scarpus vestibular. So it's going to carry information from the spinocerebellar tracts, particularly the dorsospinal cerebellar tract, synapse and Clark's column, and from the inferior olivary nucleus. So you've seen before in lab, olivocerebellar fibers. So those are going into the cerebellum through the inferior cerebellar peduncle. In terms of output, the SCP plays a key role in projecting all of that motor information back to the cortex via the VL of the thalamus and to the red nucleus. So remember, the corticospinal tract and the ruberospinal tract both were doing motor functions, coordinating particularly limb movements. So the SCP is going to be projecting to both of those structures so that they can send their messages down those two tracts to the lower motor neurons of the spinal cord. The MCP has no output, so that big, thick peduncle is only carrying information in. It's not carrying any information out. And then the ICP is going to carry that vestibular information back to the vestibular nuclei. It's also going to be sending information to the reticular formation, and the medullary, the PM is pontine and medullary reticular formation. So this is an overview. We're going to do each circuit individually, so I'll be talking about ins and outs, so you'll see this again. But you do need to know what information is being carried within each peduncle, carried in and carried out. Okay, pathways. So the cerebellum is going to have its greatest influence over motor output, over motor control, via its connections with the cerebral cortex. And it's going to be inputting, getting information, getting input from all areas of the cerebral cortex. So in this cartoon, it's only saying frontal and parietal lobes and temporal lobe. The occipital lobe will also be sending information to the cerebellum. So it's getting information about auditory information, about sound from the temporal lobe, about vision from the occipital lobe, about somatosensory integration and information from the parietal lobes, and about motor plans and motor execution from the frontal lobes. Its greatest contribution is going to be from the frontal lobe. So it's getting a lot of motor, but it's also getting sensory from a lot of places too. So this is the cortical cerebellar circuit, and I'll walk you through it, and then I'll talk about my color coding, which I'm not sure is helpful, if it's helpful or not. And I'll show a picture of it too. So we're going to go from the cerebral cortex, all of those areas that we just talked about, all four lobes. That information is going to project down to the deep pontine nuclei. In doing that, you're going to see in lab that it goes through the crus cerebri, or through the cerebral peduncles. From the deep pontine nuclei, it will travel into the cerebellum via the MCP. Remember, the MCP is carrying all of that information from the cortex into the cerebellum. It will synapse on the lateral hemispheres, or the cerebellar hemispheres, both anterior and posterior lobes, so those outside parts of the cerebellum. Deep to those lobes is the dentate nucleus. go to the dentate nucleus, and then out through the SCP to the VL of the thalamus, back primarily to the motor cortex. So its projections back are primarily to the motor cortices, areas 4 and 6, which will then send that new plan, that refined plan, down to the contralateral spinal cord, to the ventral horns and the lower motor neurons. So this is the loop, okay? From the cerebral cortex to the deep pontine nuclei is sometimes called the corticopontine tract. That's why I labeled that in green. From the pontine nuclei to the cerebellar hemispheres is referred to as the pontocerebellar tract. And then that we've already seen from the motor cortex to the spinal cord is either going to be the corticospinal or the corticobulbar tract. So the whole thing is considered a circuit, particularly going from cortex to cortex is our loop. But within that, we have at least 3 different tracks. And like I said, sometimes what's in green and blue is referred to as one whole corticopontocerebellar tract. And you can't remember which term your book uses. Probably uses all of them. Yeah.
So within this circuit, we're not looking at like first and second order neurons or where they synapse. I mean, we would be able to take out like the corticospinal, corticobulbar tract and maybe do that. But.
Right. Yeah, I'm not that concerned about first order, second order. There is a synapse in the pontine nuclei. There will be a synapse within the cerebellum, and then obviously another synapse once it gets back up to the motor cortex.
Okay.
You know, it's not like one long neuron doing all of these things.
Right.
But no, I'm not as concerned about you knowing first order, second order. I'm also not concerned, I was going to say this in lab, but I'm not concerned about laterality with the cerebellum. When we talk about function, I will talk about, you know, if you're falling, staggering to this side or falling to this side, then the lesion's probably on this side of the cerebellum. But in terms of following these tracks, I'm not so concerned about laterality of origin and termination. Okay, I don't have a picture of this one, I have a picture of the other ones. Are we okay with this? Okay, so we're going to do 3 circuits. This is the second one, the vestibular feedback circuit. The input is coming from Scarpa's ganglion and the vestibular nuclei. So the Scarpa's ganglion, obviously peripheral to the vestibular apparatus. The vestibular nuclei located within the brainstem. The cerebellum is going to get input from both. So it's getting input directly from the vestibular ganglion in the periphery and from the vestibular nuclei in the brainstem. That information is going to travel through the ICP to that flocculonodular lobe, that long, thin lobe that's sort of stretched across the whole cerebellum on its anterior surface. From there, deep to the particularly deep to the nodulus is going to be the fastigial nucleus. From the fastigial nucleus, this information is going back out through the ICP, back to the vestibular nuclei, not to Scarpa's ganglion, back to the vestibular nuclei, and then we've already seen our vestibulospinal tracts, again from 2 weeks ago, that are going to project to either extensor motor neurons in the spinal cord or to cranial nerve motor nuclei. So this is the cerebellum's way of influencing things like posture and balance and control of eye movements through its reciprocal innervation or interactions with the vestibular nuclei. So to look at that, again, in this picture, start at the vestibular nuclei. This is where our circuit is going to start. And you could note in there Scarpa's ganglion too, but for simplicity, we're going to start at the vestibular nuclei. Information travels through the ICP to the flocculonodular lobe, to the fastigial nucleus, out through the ICP, back to the vestibular nuclei, and then our pathways are going to be going either to the cranial nerve motor nuclei. Can anybody tell me what this pathway would be? MLF. Okay.
The medial.
Huh?
What's the medial?
For the most part. The ones going to the eye muscles are going to be medial from the medial vestibular nucleus, although I haven't asked you to distinguish all of those really. But yeah, going from this medial vestibular nucleus up through the MLF to the cranial nerve motor nuclei, and then down to the extensor motor neurons in the spinal cord, and these would be our vestibulospinal tracts. This is why when I get asked, like, is our final really going to be cumulative? Like, it has to be. You can't, I mean, like, this stuff doesn't make sense unless you can remember what happens after the fact. So yeah, it is integrative. Maybe not cumulative, but integrative. Okay, last circuit for the day is the reticulocerebellar circuit. So information is coming in from the pontine and medullary reticular formation. It is probably going in through the ICP, although I couldn't find any source to tell me definite. So it's a question mark there. I obviously wouldn't ask you this. It's going to project to the vermal and paravermal regions, so this would be both anterior and posterior lobes. To the vestigial nucleus, same place that our vestibular information was going. Out through the ICP, that's for sure. And back to the reticular formation in the medulla and the pons. So that's our circuit. And then we know from the medulla and the pons that we're going to have projections to extensor motor neurons in the spinal cord. And it depends whether it's coming from the medulla or the pons, what those reticulospinal fibers are doing, right? Because some are going to excite extensor neurons and some are going to inhibit extensor neurons, because those two pathways, the medullary reticulospinal tract and the pontine reticulospinal tract, work together to facilitate extension against gravity. So we don't have too much, we don't have too little. So again, this is the circuit. If we look at it here, from the pontine and medullary reticular formation, we have signals sent probably through the ICP to the vermal and paravermal regions of the anterior and posterior lobes, to the vestigial nucleus, back to the reticular formation, and then down to the spinal cord via the reticulospinal pathways. Oh, I lied, there's 4 circuits. Sorry. My bad. Last one, I promise. So this is the circuit from the red nucleus to the cerebellum. Important for this one, we have 2 structures. One, the red nucleus that we've talked about a little bit in the past few weeks. We know that that's going to be the origin of the rubrospinal tract. Where else does the red nucleus get information from? The rubrospinal tract is kind of like our backup system, or the phylogenetically older system compared to the corticospinal tract, right? So it's still very useful in animals. We don't use it quite as much. But where does the rubrospinal tract, where does the red nucleus get its information to direct motor actions? Just take a wild guess. Upper motor neurons from the motor cortex, right? So the red nucleus in this loop is going to be getting information or feedback from the cerebellum, but it's also getting information from the cerebral cortex. And then the inferior olivary nucleus, which we haven't talked about a ton even though we've looked at it in lab, is getting a lot of sensory information. And it's primarily getting information about the position and the tension of muscles. So it's getting proprioceptive information from joint receptors, muscle spindles, and GTOs. And it's going to be getting that primarily through spinocerebellar tracts. So even though spinocerebellar is going spinal cord to cerebellum, it's also going to project some of that sensory information to the inferior olivary nucleus. So the inferior olivary nucleus in this motor circuit is going to be really important as an integrative structure. So it's going to start integrating sensory and motor information even before it sends it on to the cerebellum for further integration. And what was special, this is week 2 stuff here, what was special about the inferior olivary nucleus in terms of its synaptic connections? most of our neurons in the cortex and the brainstem have chemical synaptic connections, right? Using neurotransmitters to talk to each other. But what was the other type of synaptic connection that we could have? Electrical. Remember gap junctions? They kind of just connect, they go bzz and go. It's much faster. So the inferior olivary nucleus, think back, I promise it's in your notes, has both chemical and electrical connections. And those electrical connections allowed for very rapid synchronization of signals. So again, the inferior olivary nucleus is playing a role in synchronization and coordination of sensory and motor input even before it gets to the level of the cerebellum. Once that information has passed through the inferior olivary nucleus, it will go to the ICP, to the paravermal regions of the anterior and posterior lobes primarily, to interposed nuclei, so the globose and emboliform nuclei, out through the SCP, back to the red nucleus, and down the rubrospinal tract, which we know primarily synapses on lower motor neurons in the cervical spinal cord to control upper extremity limb movements. So here it is. Inferior olivary nucleus. Sorry, this pathway starts at the red nucleus. That's my fault. The red nucleus projects the inferior olivary nucleus. Information travels through the ICP to the paravermal regions of the anterior and posterior lobes, to the interposed nuclei, back to the red nucleus, down through the rubrospinal tract to primarily flexor motor neurons in the cervical spinal cord. Good? Good enough? Yeah.
Sorry, the slide right before this, you said that the probing that comes to the inferior olivary nucleus, where does that come? Which tract is that from?
It's primarily going to come from the spinocerebellar tracts, and I'll show you that in another 2 more slides, but yeah, primarily from the spinocerebellar, which would be your dorsal spinocerebellar, your ventral spinal cerebellar, and your cuneo-cerebellar. Sorry, that's week 3 stuff. And here they are, spinocerebellar pathways. So again, this is recall. These are the 2 pathways that are carrying proprioceptive information, again, from muscle spindles, GTOs, and joint receptors. The dorsal spinocerebellar tract is primarily carrying this information from the trunk and lower extremities. The cuneo-cerebellar is an analogous system for the upper extremities. We're not going to talk about the ventral. If it's in your book, we're just going to say it's less important at this point. We'll focus on these 2. Both of these tracts will enter the cerebellum through the ICP. So this is input only. Sorry, put that in somewhere in your notes. Input only. There's no circuit here. Because again, probably the 3rd time I've said it, the cerebellum does not directly project to the spinal cord. The spinal cord will project directly to the cerebellum. So information's in through the ICP primarily to the spinocerebellum or the anterior lobe. It will also project to parts of the posterior lobe. That's the cuneo-cerebellar tract, goes to the posterior lobe. You don't need to know that. And again, some fibers will also project directly to the inferior olivary nucleus. So if we look at that in a diagram, here we're starting at spinocerebellar. Direct projections to the cerebellum through the ICP, and indirectly to the inferior olive, integration into the cerebellum via the ICP, which is the tract that we just looked at. Okay? So this spinocerebellar tracts provide that background information that I was talking about as sort of our postural set, so that even as the motor cortex is saying, this is what I want to do, it's getting information about the body, about the current position of joints, the current tension of muscles, the current extension against gravity. So it can compare what the motor cortex wants to do was sort of a where are we at in the moment, so that it can make, again, those adjustments to the motor plan before the motor cortex executes, or at least in time for the next round to be better, the next time it's executed to be better. Yeah.
What's the benefit of having direct and indirect pathways?
I guess redundancy is always good. So if you lose something, then you have a backup system for it. And they're projecting to slightly different regions of the cerebellum, so it means that the whole cerebellum is getting some of this sensory input, but it's going to get it via two different pathways. So pretty much the whole cerebellum is informed about the current position of the body before it does anything else. So I think if I had showed you this picture before class, before we went through all of that, it would have seemed really overwhelming. This is right out of your book. But it's actually pretty, you know, break it down. Our vestibular nuclei project into the cerebellum. We'll go to the vestigial nucleus, project back to the vestibular nuclei. Same thing with the reticular formation. Information comes in, it's going to synapse. Information goes back to the reticular formation. So those are two of our circuits. Same thing with red nucleus. Here's our circuit in. Inferior olivary nucleus into the cerebellum, processing by the interposed nuclei, back out to the red nucleus. Cortically, or cerebral cortex, down to the deep pontine nuclei, into the cerebellum, processing in the dentate nucleus, back out, synapse in the VL of the thalamus, to the cortex. So there are 4 circuits, and then the only other piece of that is that input is coming in from the spinal cord without any output, so it's not a circuit. I think if you go through and you study those circuits individually and then come back to this, it won't seem so crazy. It's just taking all 4 of those circuit diagrams and putting them onto one thing. The last piece of information that isn't represented in that diagram is that the cerebellum gets input from 2 other sources. We talked about the cerebral cortex, the temporal and the occipital lobes sending auditory and visual information to the cerebellum. It's also going to get auditory and visual information from the colliculi. So from the tectum, superior and inferior colliculi. And that'll project to the pontine nuclei, which is also where the cortical information is going, and then travel in with all that cortical information through the MCP. And you don't need to know the pathway, but the trigeminal nuclei that we talked about, the mesencephalic and the principal nucleus of the trigeminal nerve, trigeminal nuclei in the brainstem will also be projecting to the cerebellum. So the cerebellum has the same kind of proprioceptive input about the face, the jaw muscles, the tongue, all of that kind of stuff, that it is getting from the rest of the body. So this pathway is carrying proprioceptive touch and proprioceptive information about the face into the cerebellum, which will be important to just keep in the back of your mind when we talk about speech problems and cerebellar disorders as well. Okay, so summary of the cerebellum, its functions. We're kind of going back to big picture now. It is an integrator of sensory and motor information. It's getting a ton of information from a lot of places, and it puts it all together and makes sense of it. It compares our intended movement with what actually happened. It adjusts ongoing movement, including our postural control. It facilitates postural control, balance, and head-eye coordination, particularly through its connections with the vestibular system. It helps us to plan movement, and particularly is contributing to our ability to accurately time and sequence movements, and our accuracy in which movements are executed. And it plays a role in motor learning and motor control. So it does a lot. Again, it's the little brain, and some people think it's equally important to the cortex in terms of our ability to function. So I'm going to do a quick lab review, but any questions about lecture content? lab today, if you are able to, I'm going to try my best to orient you to the cerebellum in pictures. And there's pictures on your lab guide. But if you're having a hard time with the wet brains in particular, for me, I've already told you I'm not really good at 3D, and so it takes me a while to just, especially in the models, because they're plastic and they're, not real. But trying to figure out, like, inferior, posterior, superior, inferior, like really trying to figure out where I'm at within the cerebellum. If that is challenging, or if you need a guide, there's a good video here. You only need to watch about the first 10 minutes to really, I think it's like 26 minutes long, and they get into blood supply and stuff. You don't need to do that. But if you just feel like you need an orientation to it, see if you can pull this up in the lab or step out of the lab if you need to. It'll be worth your while. So that's posted on Blackboard under your lab stuff, or if you just want to watch it on your own after the fact. Sorry. So it is a 3D structure, and I think the easiest way when you start doing this is to orient yourself in relation to the brain stem. And I think all of our cerebellums are wet cerebellums are attached to the brain stem. When you pull them off, the models, the plastic models, you have to pull them off kind of all together. And so when you start putting them together, you're like, which way does this go? So that's why I'm talking about like orienting yourself. So if you think about it first in relation to the brainstem, the surface that is facing the brainstem is going to be the anterior surface of the cerebellum. This back part then, opposite of that, is going to be the posterior surface. And then if you look at it from above, that's the superior view. And if you look at it from below, that's going to be the inferior view. And that is the way I am teaching you, because it's the way that makes sense to me. It's the way this guy goes through it in the video, and it's the way some textbooks do it. Your book, which sometimes is a mystery to me, they talk about what we're calling anterior as the inferior view. And I assume that's because if we're talking about a four-legged animal, that would be the inferior surface. But that's really an inconsistent term, so do what you need to do to learn this, but this is how I'm going to orient you to it. So again, the surface that is facing the brainstem is going to be the anterior surface. When you look at the anterior surface, you don't need to know all of these numbers. I've labeled the ones that you need to know. This is a cartoon version of it. The way you know this is the anterior surface is that you see the cerebellar peduncles here. So in this cartoon, it's just the white stuff. Here, the big one is the MCP. Slightly below it is the ICP, which is number 4. They're both labeled number 4. I don't know why, but the blue number 4 is the MCP. The yellow number 4 is the ICP. And just medial to that is going to be the SCP, labeled number 2 in the picture. So you know you're looking at the anterior surface when you see the cerebellar peduncles. Across the surface, number 5 is going to be the flocculus, and you'll see it's kind of a thin band. It kind of gets cut off here, but it'll come all the way to the center, and this part of the center here is going to be the nodulus, or the nodule of the vermis. So this whole middle section here, 7, 8, 9 is your vermis, but on this anterior surface, we talk about the flocculonodular lobe, so the flocculus and the nodulus are in line with each other. The anterior lobe is relatively small. In this view, it's really only going to be this part here, about number 24 up. And the rest of this, 21, 22, 23, I just clicked something. Stop. Don't do it. Okay. 21, 22, 23 is going to be all posterior lobe. And then the other feature of the posterior lobe that functionally we're not going to talk about its purpose, but you'll see these big sort of hunks on the end of the posterior lobe, and those are the tonsils. It's a good anatomical landmark. So they're on the anterior surface, but more inferior. It's right here by the tonsils. And you won't be able to see the tonsils on the what brains if they are attached to the brainstem. Because think about it, the rest of the brainstem would be here, right? All right, this is the posterior view. So we see here, there's sort of this V shape, doesn't look really V-shaped, it looks more horizontal here. But it typically comes down and back up a little bit. It's a primary fissure, and that's what separates the anterior lobe, which you can see again is quite small, from the posterior lobe, which is quite large. And this big gash in the posterior lobe is called the horizontal fissure. Don't get confused, that's not what separates the anterior and the posterior lobes. It's just separating different parts of the posterior lobe. In the center again, we have the vermis. And if you put your finger on either side of that, that would be considered your paravermal region, and then everything else out here would be considered your lateral cerebellar hemispheres. This is the yeah. Yep.
Which one was the primary vision? That bottom one or the?
Nope. This. See, it's labeled here, and it goes across. They're labeling this one. You're right. I did point to this one. Yeah, and you'll see in different brains, because even that looks fairly large to me. So, in some brains, in some models, it's going to look a lot smaller. I think people get really confused, especially those cartoons where it looks like it's really big, because it's not that big. But I'm sorry, you're right. That is the primary fissure. And I'm not asking you to identify fissures. I'm just giving you the landmark so that you can try and identify the lobes today. Okay, this is a better view of the primary fissure. So again, it does make this V shape. And this is looking at it from a superior aspect. So you're looking down. Here's your brainstem. This is where the cut has been. This is your primary fissure that separates the anterior lobes. This would be your anterior lobe. From your posterior lobe is all of this. And then again, your vermis is down the center. Your paravermal region would be finger length outside of that. And then out in the edges are your lateral hemispheres. And then same thing from the inferior aspect, you are only primarily going to be seeing posterior lobe. Here are our tonsils. And in the center, set back just a little bit, is the vermis. You don't need to know the different parts of the vermis other than the nodule. It is chunked into different parts, and I'm not concerned about that. Okay. Stick with me for just a second. I thought I got rid of all these. Okay, so input from the cortex, corticopontine tract. Information is going to travel from all four of our cortical regions through the internal capsule, primarily on the outer edges of our anterior and posterior capsule, to the through the cerebral peduncles. And remember, our corticospinal and corticobulbar tracts took up the middle 3/5 of our cerebral peduncles. I know you've been dying to find out what takes up the rest. It is the corticopontine tract. So it's going to take up those outer edges of the cerebral peduncles. The synapse is going to be, we're at the level of the inferior colliculus, so we still have a little bit of the cerebral peduncle here. And the synapse is going to be starting, because we're starting to see some pontine nuclei here. So once we hit the level of the pons, our synapses are going to be in those deep pontine nuclei, which when you're looking at the black and white images are going to be the white parts, not the dark parts, because those are fibers. So the white blotches are going to be your nuclei. So up until it synapses in the pons, it's called the corticopontine tract. Once we hit the level of the pons, and again, you don't have to worry about laterality. It's on these slides. You don't have to know it. Fibers are going to cross over and go through, what peduncle is this?
Middle.
It's the middle. Here's superior. Here's middle. Nothing goes in through superior. All that motor stuff goes in through the middle. And it's going to synapse in the lateral, primarily lateral aspects of the cerebellar hemispheres. So that's pontocerebellar. Real quick, here's our inferior olivary nucleus. Again, you don't have to worry about crossings, but you do need to know that that information is going in through the ICP, also called what? It's another name for the ICP, restiform body, so that's what it's labeled in your atlas. And the olivocerebellar tract is primarily synapsing in the paravermal regions of the anterior and posterior lobe. And I may have made this a little big. Again, I told you finger length, but general region of the paravermal areas, anterior and posterior lobe. Vestibulocerebellar circuit. I've never asked you to identify specific vestibular nuclei, but they're in this region of the medulla. They're going to travel in through the ICP, synapse in the flocculonodular area, flocculonodular lobe, and information will come back out through the ICP to those same vestibular ganglia. So I don't have the output here. Dorsal spinocerebellar tract, remember Clark's column, is only going to be present mainly in thoracic vertebrae. It goes about C8 to, can't remember what we said when we did it the first time, but L2, L3 area. It's going to travel in the dorsospinocerebellar tract, which is going to be on the dorsal and lateral aspect of the spinal cord there. And that information will travel into the cerebellum. Here's running in the medulla at the level of the pyramidal decussation. Okay? And it will go in through the ICP, which is visible here, just on the outer edges. You may not have that slide. I think I cut a few out. And it will go in through the ICP, again, still at the level of the medulla. Synapsing primarily on the anterior lobe, which is also known as the spinocerebellum. Okay, a couple output slides. What nucleus is this? Oh, it's written on there. No. Dentate, okay. From the dentate nucleus, that is primarily going to, again, send out the motor information that's been processed by the cerebellum. That information will travel out through the superior cerebellar peduncle, going to the VL of the thalamus, which you saw in lab last week, because what other information goes to the VL of the thalamus? What did we study last week? Basal ganglia. So that major output of the basal ganglia it wasn't a trick question. Major output of the basal ganglia is to the VL nucleus of the thalamus before going to the cortex. Same thing for the cerebellum. Okay, the interposed nuclei are kind of this globule here. It's not covered very well, but it would be in boliform, kind of here-ish, and globose, kind of here-ish. Again, interposed nuclei is fine. That information is going to be, again, going out through the SCP to the red nucleus, which, again, you don't have to worry about laterality, but will influence the rubrospinal tract. And then the middle nuclei, the ones closest to the vermis in the center, are the fastigial nuclei. Their information is going to travel out through the ICP to the vestibular nuclei and to the reticular formation in the pons and the medulla. And you don't have to worry about laterality. Oh, my arrowheads are there. Okay. Output back to, again, from the vestigial nucleus through the ICP, back to those vestibular nuclei, back to reticular formation, and this would be at the level of the medulla, reticular formation. Okay. So in lab today, I want you to be able to identify the deep nuclei on pictures. You're not going to have to do that on the wet brains or the models. you're gonna need to look at the cerebellum from a variety of angles. So you're gonna look at those pictures in the brainstem. Make sure you can identify the different cerebellar peduncles. Think about where things are coming and going. Be able to look at the cerebellum and the cerebellar nuclei in the horizontal and coronal sections as well. You've got charts to help organize your studying in those notebooks. And then there's a quiz at the end. I did it in color because otherwise I think it would be pretty hard to identify all that stuff. But you can choose to do the quiz again on your own. You can do it today, you can do it on your own at home, or you guys can work on it together and try and get through it as a lab group. Sorry that I ran over. I know everybody probably has to pee, but we'll meet back downstairs around 10:35. Questions?